E-conference on proteomics applications to biomedical research. This conference, international conference, is organized by Vigyan Mahavidyalaya Sangola. Yes, sir, some problem is there.
Can you mute the one who is not speaking? I can hear a lot of background.
Sir, some problem is there?
Yes, sir, continue, sir. I will check.
Okay, sir. Okay, sir. This is the second day of the international conference, and today's speaker is Dina Samodova from Denmark. Here, the responsibility of introduction of is given to me. So, Dina Samodova, she has completed her PhD starting from initially in 2015. She has completed Bachelor of Natural Sciences from University of Latviia, Faculty of Chemistry, Riga, Latviia, and her bachelor's thesis was photostability of nitrofurazone solutions. She has then completed internship in bioanalytical chemistry, where the project title was simultaneous determination of active pharmaceutical ingredients in water samples using one-line solid phase extraction LCMS method. Then she has completed MSc in the chemistry from University of Latviia, Faculty of Chemistry, Riga, Latviia, and the thesis of her master's degree was the studies on anti-radical activity kinetics of the different antibacterial plant extracts. Then she has done second internship in Proteomics from University of Bordeaux, Center for Functional Genomics, Bordeaux, France, and she has completed her PhD in molecular mechanisms of disease from University of Copenhagen, Nordics, Foundation Center for Protein Research, Copenhagen, Denmark, and the title of her thesis was Mass Spectrometry Technology for Paleoproteomics. She has work experience as a laboratory assistant, analytical chemistry teacher, laboratory assistant in liquid chromatography, mass spectroscopy, then as a PhD candidate and a postdoctoral researcher at University of Copenhagen, Denmark. Then she has teaching experience at various levels as she worked as actually in a teaching field as flashback tutorial, bridge translational discovery omics 1, advanced methods of the analysis of protein disease mechanisms, then bridge translational discovery omics 2nd, MS Frager and DIANN tutorials and master project co-supervision. She has peer review experience as a Review Commons and Journal of Proteomics, which is independent peer review before journal submission. She has received fundings from actually she has two types of the scholarships: teaching emerging methods in paleoproteomics for the European research area, that is European Training Network, in a Marie Curie ETN European Training Network, provided a doctoral training in paleoproteomic analysis in Europe's cultural heritage materials. And she has also received second fellowship from Enhancing Human Capital and Knowledge in Health Science by Institutional Cooperation and Mobility between the University of Latviia and 3 Norwegian universities. She has some outreach work as she worked as a part of the local organizing committee focused on outreach activities, interaction with various students, teenagers and adults also. Apart from this, she has several publications in the reputed journals like Molecular Cell Proteomics, Springer Nature. Also, she has published 2 papers in Nature whose impact factor is more than near about 42. And then Frontiers in Chemistry, American Chemical Society, Crystal Growth and Design, Journal of Pharmaceutical and Biomedical Analysis, Science Direct elsewhere. So, which are having very good impact factor. So, with this brief introduction of Dina Samodova Madam, I welcome Madam and request her to deliver her lecture on mass spectroscopy technology for proteomics. Dina ma'am, it's over to you now.
Thank you very much for an explicit introduction, and good afternoon, everyone. I will now share my screen and start the presentation. So I hope that everyone can see my screen well.
Yes,
thank you. So I'm really grateful for this opportunity to present at this event, and I hope my talk will be useful and interesting. So as you can see from the title, I will talk quite a lot about mass spectrometry technology today. And since we don't really know about your background in proteomics, I will go into details and try to make it as simple as possible. However, if you have any questions, please don't hesitate to stop me and ask. So first of all, I would like to walk you through the concept of proteomics, and then we will move to the more technological part of the talk, which embraces liquid chromatography technique, mass spectrometry, and data analysis. And I will finalize the talk by speaking about a couple of recent advancements in proteomics. So why are we interested in proteomics? So when we look at these two organisms, the caterpillar and the butterfly, we can obviously see that they're completely different. However, the interesting thing is that they're sharing the same genome. However, the proteome of these two species is completely different. So to understand how is that possible, we actually have to learn what proteomics is. So proteomics is actually protein complement expressed by a genome. And proteins are basically translated from RNA, which is in terms transcribed from the DNA. So during the transcription process, genome is undergoing a round of biological processes, such as, for example, alternative splicing, which results in the expansion of the transcriptome. As you can see, we start with the 22,000 genes, and during the transcription process, it expands to 200,000 transcripts. Whereas when RNA is being translated into proteins and a range of post-translational modifications is taking place, then the proteome, the final proteome, which we obtain at the end of this translation, is actually larger than 1 million of proteoforms. And this is actually answering our question, why is the caterpillar different from the butterfly? So basically, this different organisms may have the same genome by completely different proteome because it's undergoing a range of modifications during the transcription and translation process. So to study proteins in the biological samples, we use the analytical technique called proteomics. And it can also be quite challenging to analyze proteins in biological samples. So first of all, because proteome contains proteins of the completely different concentrations. As you can see here, the dynamic range of protein concentrations can range from 1 to 10 E8. It also contains proteins of completely different sizes, which can be either very small proteins or very large proteins, with completely different isoelectric points, hydrophobicities, and also stabilities. And finally, tissue-specific protein expression levels can also be completely different. So these are the main challenges of proteomics. And however, even though it still possesses a lot of challenges, it also has a range of various applications. So first of all, proteomics is used for general protein analysis within the cell or in different tissues, or to compare protein expression levels in different conditions. It can also be used to characterize protein localization within the cell. Proteomics can be used to study protein interactions, as well as the post-translational modifications, which I have mentioned in the previous slide. So post-translational modifications are actually quite important because they can alter the biological function of the proteins. So that's why it is also quite important to study these kind of modifications by means of proteomics. So in this time scale, you can see how proteomics emerged since the end of the 20th century. And this was basically possible thanks to the development of various analytical techniques, such as mass spectrometry, which evolved already in 1970, gel electrophoresis, nucleotide sequencing, and various other analytical techniques. However, the real development of proteomics technique has only started in the 21st century, or in the year of 2000, when tandem mass spectrometry was invented. So in common bottom-up proteomics pipeline, which is basically used to analyze proteins in our biological samples of interest, we firstly have to extract proteins from the biological sample, which can be either cells or any kind of tissue, for example, blood plasma samples, even bones and teeth, or muscle tissue or adipose tissue. So when proteins are getting extracted, we have to unfold them or to denature. So this can be done either using high salt or high temperature, and the proteins are being unfolded, and the disulfide bonds, which are holding the protein structure together, are getting reduced and alkylated with the corresponding chemicals. This is required in order to further cut or digest proteins into shorter amino acid sequences called peptides, which are smaller units of the proteins, and they are more amenable for mass spectrometric analysis. Since proteins are large molecules, it can be quite challenging to sequence them directly by mass spectrometry technology. So that's why we have to follow all these complex preparation protocol and cleave or digest proteins into shorter amino acid sequences or peptides. So after our peptides are being sequenced with the mass spectrometry technology, we pursue the data analysis. And now I would like to talk a bit more about the actual technological core of this entire proteomics pipeline, since it is very crucial, and without this part, no protein analysis with mass spectrometry-based proteomics would be possible. So first of all, I would like to introduce you to the liquid chromatography analytical technique, which is basically a method of molecular separation based on their hydrophobicities. So the analytical capillary column is typically packed with the silica-based material beads, which are modified, and in proteomics, we're typically using C18 or octodecyl modified silica sorbent material. So this sorbent material in turn allows to basically separate peptide molecules based on their retention on the sorbent material and elute them in portions from the analytical column using different percentage of acetonitrile. Why is it necessary to use this technique in proteomics? So as I mentioned in the previous slide, proteins and peptides are quite complex. And imagine if we have a mixture of highly abundant red peptides. As you can see in this slide, I have different colors of peptides being separated on the column. And then we have also low abundant yellow peptides. So if we directly infuse them into mass spectrometer, the mass spectrometer will of course sequence only the most abundant red peptides, and it will completely miss the yellow peptides. So based on this, we have to separate these molecules prior to MS sequencing, and liquid chromatography is used exactly for this purpose. So it is used to reduce sample complexity prior to MS analysis. And in this slide, you can also see various technical parameters, which are quite complicated, and they can be basically used to ensure highly efficient chromatographic separations. This is just an example setup, which is typically used in bottom-up proteomics to separate peptides, and it is called EASY-LC liquid chromatograph system. So it contains a series of high and low pressure pumps, which allow to deliver the sample with a high pressure pump to the analytical column, as well as low pressure pumps are being used to deliver the solvent, which is acetonitrile and water, in order to elute our peptides from the analytical column into the mass spectrometer. So after our peptides have been successfully separated using the liquid chromatography technique, they're actually ready to be sequenced by mass spectrometry. So mass spectrometry is an analytical technique which measures mass to charge ratio of the different ions. So it's basically a kind of scale. And the more heavy ions have higher mass to charge ratio and vice versa. Mass spectrometry can be used for multiple application areas, such, for example, as the quality control of organic synthesis, for metabolomics analysis, for proteomics, and also for forensics, for example, food chemistry, and many, many other application areas. However, there is one major drawback which mass spectrometry possesses, and that's the discrimination between structural isomers. So for example, here you can see the MS or mass spectrometry spectrum of pentanone. And as we know, both 2-pentanone and 3-pentanone, which are the structural isomers, have the same molecular weight. So as a result of mass spectrometric analysis, a mass, excuse me. Hello.
Can you mute Dipali Patil? Can you mute the microphone, please? Yeah.
Okay, so I can continue. So basically, yes, as a result of mass spectrometric analysis, we obtain a mass spectrum of this pentanone molecule, which has mass to charge ratio of 89. However, since both isomers are having same molecular weight, we cannot really discriminate between the two structures using this method. To do so, we actually have to use tandem mass spectrometry. So tandem mass spectrometry includes a secondary fragmentation event of our analyte molecules, which allows to generate the fingerprint mass spectra of a particular molecule. So this way, it allows for peptide identification and also for structural isomer discrimination. So tandem mass spectrometry consists of the two scan events. So the first scan event is called MS1, or just MS spectrum, and it's virtually same as just MS or mass spectrometric analysis, which doesn't allow us to discriminate between the structures. However, on the MS2 level, the fragmentation of molecular bonds is happening, and as a result of this fragmentation, we obtain unique fingerprint mass tandem mass spectra or MS2 spectra of particular molecules. So this way we can actually discriminate between 2-pentanone and 3-pentanone because, as you can see, they have completely different fingerprint MS spectra. So what about peptides? So well, in case when we have two different peptide sequences with completely different molecular weights, it is even possible to discriminate between these two molecules on the MS1 level. As you can clearly see here, these two peptides have mass to charge ratio of 782 and 841. And this way it's possible to discriminate between them even just on the MS1 level. However, what do we do in case when we have multiple peptides sharing the same molecular weight? So we can't really discriminate between those using just MS1 or mass spectrometry. So for this purpose, we again have to use tandem mass spectrometry and generate their fingerprint mass spectra or MS2 spectra. And when we fragment the two previously mentioned peptides, we obtain unique ion series spectra corresponding to each of the peptides, which in turn allow us to discriminate between these two peptides on MS2 level. So why do we actually need a MS1 analysis in proteomics if we are identifying peptides on the MS2 level? Well, MS1 level is typically used for peptide quantification in proteomics. However, I will not go into too many details about this in today's lecture. So now I would like to introduce you to the actual schematics of the real mass spectrometer, and this is just a schematical drawing which contains the major parts of each mass spectrometer. So it's the ion source, the mass analyzer, and the detector, which are the heart of mass spectrometer. So it is important to ionize peptides prior to a mass analysis because we want to allow them to fly inside the mass spectrometer and to be analyzed. And for this purpose, two soft ionization techniques such as electrospray ionization and matrix-assisted laser dissociation can also be utilized. However, MODI is less commonly used nowadays because it's just suitable for ionization and identification of one or several proteins. So it's very simple, it's very cheap. However, it cannot be used for complex peptide mixture analysis, and it cannot be coupled to the liquid chromatography systems compared to the electrospray ionization. So that's why electrospray ionization is #1 ionization technique in proteomics. So electrospray ionization can be done in positive and negative modes. And for proteomics analysis, we typically use positive ionization mode. And this is because of the structure of our peptide sequences. So any peptide contains the C-terminal and the N-terminal. And N-terminal typically includes the amino group, which is a basic residue, and can attract proton as a result of positive ionization. Whereas the C-terminal of peptides contains basic amino acid residues such as arginine and lysine, which are also eager to attract proton as a result of ionization in positive mode. Whereas, for example, if we look at the lipid molecule, which does not contain any basic functional groups, it is virtually impossible to ionize this molecule in positive mode. So that's why we have to use negative ionization mode in this case, because hydroxyl group is more eager to subtract proton rather than attaching the proton. And when the ionization has happened, the peptides enter mass spectrometry and are directed inside the mass spectrometer using this series of lenses. And afterwards, they follow inside the mass spectrometer through the advanced active beam guide, which actually filters out neutral molecules. Further on, peptides are getting filtered using the quadrupole mass filter and fragmented in the high energy collision cell. So this high energy collision cell, or HCD cell, as you can see in the drawing to the left, is a unique place where peptides collide with the neutral gas molecules, such as, for example, argon, nitrogen, or helium. And as a result of these collisions, peptide fragments are generated, which are further detected in the Orbitrap mass analyzer. So this is a schematics of the most commonly used Orbitrap mass analyzer. And as a result of this fragmentation, the MSMS spectra are generated. So these are basically same MSMS or tandem mass spectra, which I introduced to you in the previous slides. So peptide fragmentation typically occurs across the peptide bonds, which are the weakest bonds in a peptide molecule. And basically using different fragmentation techniques is possible to generate completely different ion series. However, collision-induced dissociation, or CAD, or also HCD, such as higher energy collision-induced dissociation, are the most commonly used fragmentation techniques in proteomics. And this is because they generate nice and intense fragment ions, which can be used for simple peptide identification. So in collision-induced dissociation, our ionized peptides enter the collision cell, where they collide with the neutral gas molecules, and as a result of these collisions, charged fragments, neutral fragments, and also secondary fragmentation events are occurring. So in collision-induced dissociation, a range of molecular rearrangements is leading to the formation of two intense ion series, which are Y ions and B ions. which are highly suitable for peptide identification by tandem mass spectrometry. Another fragmentation technique, which is also sometimes used in bottom-up proteomics, is called electron transfer dissociation, or ETD. And this is the technique where an external source of electron donor is required. So in this method, which is followed again by a series of molecular rearrangements, two ions, one of those is cation radical or Z ions, and the other one is just cation, C ions, are generated. So these ions are less intense than Y and B ions, and that's why electron transfer dissociation is not a technique of choice #1 when it comes to bottom-up proteomics. However, it can still be useful for multiple specific proteomics applications. And finally, you can see a summary of different kinds of mass spectrometers available on the market, such, for example, as time-of-flight mass spectrometer or quadruple mass analyzer, as well as ion trap, and so on and so forth. However, as I already mentioned previously, the Orbitra-based mass spectrometer is typically the most commonly used mass spectrometer nowadays. And this is because it is highly sensitive, it possesses high resolution and high sequencing speed. So finally, when our peptides have been separated by liquid chromatography and sequenced by mass spectrometry, we can obtain our peptide identification list or protein lists. So protein identification can be pursued using the two major data analysis approaches, such as database searching and de novo sequencing. So I will tell a bit more about database searching in the next slide. However, I must say that database searching includes reference protein database, but in case when we're dealing with, let's say, novel proteins or monoclonal antibody analysis, for example, where the sequence of the molecule is unknown, we have to perform de novo sequencing or database independent sequence reconstruction of the molecule by inferring peptide sequences directly from high quality MSMS spectra. Furthermore, it's also possible to pursue data analysis of post-translational modifications in our proteomics samples, which is typically done using either open search data analysis approach or data-dependent peptide search, which are both quite complex. So now coming back to the most commonly used approach for peptide and protein analysis called database search. So in the database search, a reference protein database of the corresponding species is in silico digested to obtain in silico MSMS spectra of the corresponding proteins. And these in silico MSMS spectra are matched or compared to our experimental MSMS spectra, which were generated experimentally by fragmenting the peptides inside mass spectrometer. So the goodness of fit between the in silico generated MSMS spectrum and the experimental MSMS spectrum is evaluated with a so-called score. And the peptides with the highest goodness of fit or with the highest score are typically assigned as the best match. And this is how we end up with our identifications. So after peptides are being identified, they are being inferred to the proteins. And at the end, we obtain a list of protein identifications inside our biological sample of interest. So now that I have gone through the typical bottom-up proteomics pipeline, I would also like to tell you a couple of words on the latest advancements in the field. So of course, if we want to achieve high sensitivity, high speed and throughput of our proteomics analysis, we are in need for automated workflows. And these can be enabled using the latest robotic systems, such for example as Kingfisher robot, which can be used for sample preparation, and in combination with the latest liquid chromatography mass spectrometry techniques, these can lead to ultra-fast peptide identification in just 14 hours of the instrument time. And these can be virtually used for any kind of tissue, as you can see from the figure B. Moving further, we are of course in need for fast and robust peptide separation prior to MS analysis, and this can be done using the novel EOSEP-1 liquid chromatography system, which is quite different from the nano-LC, which I have shown to you in the introduction slides. And the main difference is that it actually contains a dual low pressure pump series, which allows for higher mobile phase flow rate and also removes the overhead injection time for the sample. So by using this liquid chromatography system, we can achieve a robust and high throughput, high flow rate, higher mobile phase flow rate peptide separations. Moving forward, we are also interested in multifunctional MS sequencing. And for this purpose, the mass spectrometers which contain multiple mass analyzers are implemented. One of those examples is the Eclipse Tribrid mass spectrometer from Thermo Fisher Scientific. And this mass analyzer is quite unique because it contains the quadruple mass filter, the ion trap mass analyzer, and also the Orbitrap mass analyzers. So it's basically a hybrid which contains 3 different mass analyzers within one setup, thus allowing for orthogonal mass fragmentation or for multiple fragmentation modes of the peptides. It also has a range of series of features which improve peptide quantification. And finally, we also need high sensitivity peptide sequencing, which can be enabled by a new timsTOF Pro system from Bruker. And this mass analyzer contains the so-called trapped ion mobility spectrometry unit, which in turn allows for peptide separation both in mass to charge and also in the ion mobility dimensions, which in turn increases the efficiency of ion utilization. This type of mass spectrometer also allows for the event called parallel accumulation serial fragmentation, which allows for simultaneous fragmentation of multiple ion series. So it basically increases the sequencing speed without the loss of sensitivity. So these were the main, advancements in the field of proteomics, which we're currently using in our daily activities. So finally, as we can see, the field of MS-based proteomics has already achieved quite standards of development. However, I personally believe that there is always a need and a room for further technological improvements, because we're always looking for higher speed, higher sensitivity, and higher robustness of our measurements. With that, I would like to thank you for your attention, and I'm ready to answer your questions.
Hello. Hello.
Hello.
Yes, ma'am. Now, participants are requested to raise any questions or queries if they have. Now, the session is open for discussion.
Yeah.
Dasani, sir.
Hello, sir. Bola.
Yes, sir. Anybody has any question, then they may ask.
If there are no questions, please conclude, sir, session.
Okay, sir. Ma'am, Dinah, ma'am.
Yes.
Can you explain regarding some doubly charged ions? Why there is doubly charged ion formation in peptides?
Yes, sure. Actually, then I have to go back to my presentation. I'll just show you the slide. So in bottom up, of course, I haven't mentioned this because of the interest of time, and also because we are, we wanted to make it simple. So in bottom up proteomics, we're typically using a protease called trypsin. So at this step, when we have to digest our proteins into peptides, we have to use the protease. And the protease of choice is typically trypsin, since it's highly efficient and highly aggressive protease, and also very specific. So it generates the ions which contain arginine and lysine at the C terminus. And going back to the ionization figure here, this is why we actually get the doubly charged peptides. So all peptides always contain a basic residue, the amino group at the N terminus. So of course, that's why it attracts the proton during the ionization, as well as when we use trypsin, this residue or R is corresponding to arginine and lysine, which are in turn basic amino acids, which also attract proton. So I hope that answered your question.
Yes. So, Tombre sir, can we conclude now?
Yes, sir, conclude.
Okay. Dear participants and organizers, Dynamam has very explained in a very simple way, fingerprint mass spectrometer, difference between peptide and dipeptides, Marley salt ionization techniques for peptides, modes of ionization and fragmentation patterns. Also protein identification, peptide separation, and peptide sequencing related things. So on behalf of organizers, Department of Chemistry, Vijnan Mahavidyalaya Sangola, I would like to thank Laina ma'am. Thank you very much.
Thank you. Have a nice day.
Thank you.
Thank you, Dr. Devkar sir, for valuable discussion and handling the session. Also, my sincere thanks goes to Dr. Diana ma'am for her nice presentation. Now we will move to session 6. Once again, I welcome all in session 6. The session coordinator is Dr. Sheetal R. Shinde ma'am. Dr. Shinde ma'am, it's over to you.
Yeah, I will start my.
Thank you, sir. Good afternoon, everyone. I am Dr. Sheetal Shinde, Assistant Professor in Vijnan Mahavidyalaya Sangola. I heartily welcome Honorable Dr. Batsute sir as a chairperson in International E-Conference on Proteomics Application to Biomedical Researcher 2022. I would like to introduce Dr. Batsute sir, Principal, Sangola College, Sangola. Here, chairperson for LIC committees, affiliation for various programs, research laboratories, Punyashlok Ahilya Devi Holkar, Solapur University, Solapur. He is member of selection committee for appointment of principals in various colleges, member of Senate, member of board examination, member of BOS in chemistry and academical council in PAH Solapur University, Solapur. He is coordinator IQASC in KBP Mahavidyalaya, Pandharpur. Life member trajectory journal published by RIRD Satara. He participated in number of orientation and refresher courses in different colleges and universities. He has published a number of research papers in well-reputed national and international journals with higher impact. He has completed various UGC research projects, while some projects are ongoing. He is a resource person in various academic section, green chemistry approaches, Indian Constitution and social justice. He has presented more than 50 papers in national and international conferences. He has honor of research guide in chemistry, PAH Solapur University, Solapur. Under the guidance of Dr. Batsute sir, 3 students awarded PhD degree and 1 student completed MPhil degree. He is awarded as an ideal teacher in Punjab Rao Deshmukh Rashtriya Shikshan Parishad Pune division. Also, best teacher in Raya Shikshan Samstha's KBP Mahavidyalaya, Pandharpur. He plays extension activities in various institutions and universities. He also attended more than 50 national and international conferences and workshops. Respected Dr. Batsute sir has very nice and fruitful academic data. I am very much thankful to Dr. Batsute sir for kind our national e-conference. Thank you, sir.
Am I audible?
Yes, sir. It's audible.
Am I audible? Hello.
Yes, sir. Yes, sir. It's
Okay.
audible.
Okay. Good afternoon, everyone. Department of Chemistry of Vijnan Mahavidyalaya, Sangola, on initiation of IPSC, has organized the two-day e-conference on proteomics application to biomedical research. So on this occasion, the present honorable members of the management of Vijnan Mahavidyalaya Sangola, Principal Dr. Raghunath Phule, Resource Person Dr. Atul Deshmukh, Convener Dr. R.S. Gaikwad, Organizing Committee members, teachers, researchers, students, I welcome you all in this session of the second day. I am here to introduce Dr. Atul Deshmukh, who is working as scientist in University of Copenhagen in Denmark. Denmark is a very happy nation. As you know that on 18th March 2022, United Nations have released the report on the happiness index of the nations of 146 nations, and out of these 146 nations, the Denmark ranks second. And therefore, he is very fortunate to work there as scientist. For students and researchers, it would be very encouraging to know that the boy born in Sangli, after completion of his BS degree in pharmaceutical sciences in 2000 from Pune University, went to Sweden and completed MSc in molecular biology and biotechnology from a very reputed institute there. He obtained PhD in nutrient and energy sensing in skeletal muscle from Karolinska Institute, Stockholm, Sweden. His most of the research work is on very hot topic today, that is obesity and diabetics. I am very happy to inform you that he has fetched grant of 5,53,000 euros during the period 2007 to 2019 for completion of PhD, postdoc, and different research projects related to proteomics. At present, 3 research projects related to diabetes worth 2,68,000 euros are ongoing under his guidance. As far as his research interest is considered, he is working in diverse fields of proteomics, that is application of mass spectrometry-based proteomics, skeletal muscle metabolism, exercise physiology, mitochondrial proteomics, secretomics, peptidomics, obesity, type 2 diabetics, post-translational modifications. He has worked as supervisor also for bachelor, master, PhD students and research assistants. As a leader, to develop leadership, he has completed 4 leadership courses, and he is able leader now, working from 2014 to 19 in Novartis Institute, and he is a teacher also. He has worked as tutor and supervisor also. As far as his research publications are considered, during his journey from PhD to scientist, he has published 35 research articles, out of which 32 are peer-reviewed and 3 are reviews. I would like to inform you that his citation index is 3,143 and H index is 23. So very good work he is doing in the field of research, and he has worked as a referee for reputed journals. So with this brief biodata, I welcome him again and request him to give his presentation. Thank you, sir.
Okay, thank you for kind introduction. I will share my slides now. Can you see my slides?
Yes, sir.
Okay, so yeah, very happy to present at this meeting. So I actually come from the same region. So I'm originally from Wangi, which is not very far from Sangola, close to Temborni. So like मी मराठी माणूस आहे आणि सर्वसाधारण बॅकग्राऊंड मधला मराठी माणूस आहे. गावाचं बॅकग्राऊंड आहे. So even though you have introduced me, I think I would like to give again the brief introduction. I have one slide about my scientific journey. But before that, just an intro slide. So currently I'm working at Copenhagen University. Copenhagen is the capital of Denmark. And within this university, I work at this center called Center for Basic Metabolic Research, and is funded by Novo Nordisk Foundation. And my title today is a group leader and associate professor. So we don't do any teaching, but we do full-time research. And I have a team of 10 people now within the group, and we mainly work with diabetes and metabolism related diseases, and we apply proteomics technology to study this thing. Yeah, before I go into the science, I can give you a little bit background again. Like, you know, I'm a pharma graduate. I studied in Bhor, you know, like RDCOP College Bhor, close to Pune. After finishing my graduation, I worked as a teacher there on the Diploma Pharmacy College, and I was preparing for the different entrance exams. Then in 2004, I came to Sweden, where I studied masters in biotechnology, which is completely different than pharma education. So it's a lot about DNA, RNA, and the molecular biology. Then I did a PhD at Karolinska Institute, and the Karolinska Institute is the institute that gives a Nobel Prize in the medicine. So it's quite famous for that. And I was working there for 6 years almost. And I also want to mention that the PhD in the European country or Scandinavia, it's very different than the PhD that we do in India. Here, the PhD is considered as a normal job, like you pay taxes for your salary and you get the normal salary that you can get it as a bank worker, for example. So it's a very different. And also, in order to finish the PhD, you need to have a certain number of publications here, and that's not the criteria in many different countries. So after PhD, I had a fellowship to go to Brazil as a visiting researcher, where I worked on the some aspect of brain and its role in metabolism. And I had an opportunity to work there for long term, but I was not interested because I had another job offer, and that was at the Max Planck Institute Munich in Germany, where I had a fellowship. I stayed there for 4 months, and that's where I started to learn about proteomics. Then I came back to Denmark. I came to Denmark for a short period of postdoc. I became associate professor in year 2016. And since 2019, I have a group here at the Copenhagen University. And today, even though I work in Denmark, I'm living in Sweden. So this is a bridge between Denmark and Sweden, and we are seeing from the Danish side. So I live on the other side of Sweden. Even though these are two different countries, it takes only 1 hour for me to get from home to Denmark. So yeah, I live in Sweden and work in Denmark. Yeah, so a little bit about our institute. So if you look at the Google map, we are located here. It's a city center of Copenhagen. And this is a building where we are located. It's a very beautiful modern building, and we are located on the 8th floor. And the name of our center is Novo Nordisk Foundation Center for Basic Metabolic Research. We can see more about this center on this website, or you can also look at the link of my group. And it's a very important thing to mention that we are funded by Novo Nordisk Foundation. It's a non-profit organization who gets money from their companies. They have two big companies. One of them is Novo Nordisk, which is the biggest producer of insulin. So like if you look at the insulin market in the whole world, they have the largest share. And whatever profit they make from the company, it goes to foundation, and foundation puts a lot of money in the research. And that's why our center is also called Novo Nordisk Foundation Center for Basic Metabolic Research. Yeah, so then I will move to the diabetes as a problem because my research is quite related to diabetes and obesity, and I think everyone in the crowd should know that diabetes, it's a big problem. It's a huge problem, particularly in India. It's getting really big problem. So there is this organization called International Diabetes Federation. Every year they make a report, they make a statistics, how many people are suffering with the diabetes. And their report is quite alarming. Like you can see, there are approximately 537 million people in the world that are suffering with the diabetes. And in addition to that, these numbers are gonna increase to 783 million by 2045, which is quite scary. And today, like in the low or a middle income countries like in India, 3 out of 4 people are living with diabetes, but they don't get diagnosed. And it's a huge burden on the healthcare system. That's why diabetes actually it's even bigger problem than COVID. But you just don't know the seriousness of it because things happen quite slowly, and people don't recognize how dangerous it is until you get serious problem. Also, you need to remember that there are 2 types of diabetes, type 1 and type 2. And type 1, it's a problem, but it's not a major problem. But the type 2 diabetes is a major problem. And the biggest difference there is in type 1 diabetes, your pancreas are not producing insulin. And that's why you are completely dependent on insulin and you need an insulin shot to survive. But in case of type 2 diabetes, it is mainly because we are not moving so much, we are eating too much, we have a bad healthy lifestyle, you get this belly, you get obese, and then your blood glucose level is quite high, and that increase in the blood sugar level can cause many different problems to different tissues and many different complications. And my work is mainly on the type 2 diabetes because the prevalence of type 2 diabetes is quite high. So a little bit background about obesity, diabetes, and the sedentary lifestyle. So as you know, a lot of people these days, they don't want to walk, they don't want to run, they just want to sit home with their next to smartphones, or they want to eat more high, like fried food, oily food. And that's why there is this something called diseasome, meaning that if you're physically inactive, if you're not moving so much, you get all these different diseases, cardiovascular disease, type 2 diabetes, obesity, depression, dementia. So this is a term that they call it diseasome. It's not only one disease, you get many different disease just because you have a bad lifestyle. It's just that because you're not moving enough and you're eating too much. And that is a major cause for diabetes. So what happens in diabetes is like you have high blood glucose level, and this term is called as hyperglycemia. And when you have a diabetes, you have a failure in many different organs, including muscle, liver, adipose tissue, brain, like pancreas and everything. So that is what is depicted here. And when doctor diagnosed you as a diabetic, it is already too late because the process has been started many years before. And that's the reason why diabetes is very difficult to treat. And you can see here, like I have a small square that are highlighted. These are different drugs that are used for the treatment of diabetes, meaning that diabetes cannot be treated by one drug. You need multiple drugs because these drugs act on different organs. You have a multi-organ failure. On the other side, if you do physical exercise, little bit of walking or running or swimming or biking, you have a lot of beneficial effect on the diabetes. So all these things that I have in the previous slide, that you have multiple organ failure, you have a beneficial effect on these organs if you do a little bit of exercise. The exercise doesn't need to be running and lifting weights, but it could just be like 30 minutes of walk every day, because exercise has a beneficial effect on all these different parameters in different tissues. And in fact, if you look at the population studies, you can see like the best used drug, which is metformin for the treatment of diabetes. That works quite well for the treatment of diabetes. These are incidents of diabetes. But in addition to that, if you just manage your lifestyle, eating well and moving much, like quite a lot, then you can increase your you can decrease your incidence of diabetes just by managing the lifestyle. So the message here is like physical exercise and also diet are the essential component of treatment of diabetes and obesity. So I work quite a lot with muscle, and the reason for that is muscle is one of the largest organ in our body. 40% of our weight is the muscle. And on top of that, muscle is a major depot for glucose. So when you stand, when you move, you need energy, and that energy is mainly produced from the glucose that is in your muscle. So in this figure, you see when you what this shows, like this is a technique called clamp, where you can see after you eat the meal, after you eat the food, where does the glucose goes. So the yellow part mentions the portion of glucose that goes in the respective organ, like that is muscle. So 80% of glucose after our meal goes in the muscle, but when you have a diabetes, this is severely impaired, meaning that once you have a diabetes, something happens to muscle and it's unable to take the glucose inside, and that's why you have an increased level of sugar in the blood. And this is basically what happens in diabetes. And if you go in the molecular level, now this is a diagram of the muscle. How does the glucose goes in the muscle? So this, imagine that this is a glucose in the plasma. There is this transporter called GLUT4. So when you have insulin stimulation or when you do exercise, you activate many different kinds of proteins, which is on the left and the right. And this activation leads to migration of this GLUT4 vesicle from cytoplasm to plasma membrane. And that's where glucose comes in. And when you have a diabetes, this pathway that I highlighted here in the red, it's impaired, it doesn't work. And this is a condition that is called as insulin resistance. Even though you have an insulin in your plasma, it's unable to work efficiently. That's why you don't have so much glucose inside your body, inside your muscle. And then that's why you have increased level of sugar in the plasma. So in my group, which is called as functional proteomics and metabolism, we hypothesize that sedentary lifestyle and unhealthy eating leads to development of obesity, while if you eat well and if you do exercise, you have a healthy lifestyle. And we hypothesize that these changes are mediated by proteins in different organs. And that's what we study by mass spec. And then the various aspects of protein dynamics, I call it, are this protein-protein interaction, post-translation modification, protein secretion. So I will highlight some of these aspects. So Diana nicely explained you how proteomics technology work. So proteomics is a technique where you use mass spectrometry to measure abundance of many different proteins at the same time. And you start with the sample preparation, like the sample could come from any tissue or biological fluid, and you digest it to the peptide, you separate peptides on the on the LC, and you measure on the mass spec, and using the database approach, you get the list of protein ID. And once you know what is up and down in certain condition, you could do the functional follow-up and study that particular protein in detail. So I will go now to the muscle proteomics. So the muscle, this is how it looked like. It has this hair-like small fibers that are bundled together, and it looks like a rod, literally like a rod, but they are very tiny. And muscle is very, very complex organ. It has multiple different type of cell types inside, and it has a unique contractile and metabolic properties. And the way we study muscle is we either take the muscle biopsy from the diabetic patients or, from the mouse model, or we also do the cell culture. But to study the proteomics of muscle is very challenging because the difference between the highest abundant protein and the lowest abundant protein in the muscle is wide, and it's proteomically very, very challenging. And without going in detail, I can mention that I have worked extensively for last 10 years to make the methods more efficient to measure maximum number of protein possible in the muscle. So this is, again, I don't know how much you're going to understand this, but this is one of my first technical paper where I developed different different fractionation methods, where we fractionated peptides into 12 different fractions, and we use some computational approaches to map a deep muscle proteome. And then there is this algorithm called match between run that we use for matching. And the message here is like we managed to detect 8,000 muscle proteins, and this is still the largest muscle proteome. And this paper is kind of highly cited paper. It has more than 200 citation now in 5-6 years. So what this paper shows that the proteome of the muscle cells and the muscle tissue is very different. The cells are less complex and the muscle tissue is more complex. And this just shows the dynamic range in these two different tissues. So yeah, this method was very good, but it was time consuming, and this method was not really ideal for the large scale studies. So we came up with another method where we do the digestion of protein using different enzymes when we collect the fractions. And compared to the previous slide, here you have few fractions and you measure one sample fewer times compared to the like let's say 12 times. And you get increased sequence coverage with a short measurement time. We applied this method to study muscle proteome of the diabetic mouse model, where we use only 2 fractions, and we could show that the mitochondrial protein was significantly affected. And this is a volcano plot that shows which proteins were upregulated and downregulated in the diabetic mouse. And we revealed quite a lot of new information in this one, and it was published in the Proteomics journal. Then I would like to move to like 2 muscle types that we have. So if you look at the weightlifter, they have a big muscles, and if you look at the marathon runner, they have small muscles, right? But inside, if you look under the microscope, the marathon runners, they have so-called slow muscle fiber type, which has more mitochondria. So this dark part that you see here is the mitochondria. And you can see that if you are weightlifter, you have less mitochondria. And metabolically, these tissues are very, very different. So it is good to have more these slow fibers because then you can run long, you have a healthy lifestyle. So in one of our projects, we recruited 5 healthy individuals. We took the muscle biopsies before and after exercise, and we isolated the slow and fast fibers, and we looked at their effect on the exercise training. And this graph just shows that the exercise training, which was for the 12-week, had worked very well. They increased their exercise capacity, which is measured by VO2 max. They increase their ability to handle glucose and the glycogen. And what we show, this paper was published in Nature Communication. What we show here is like the slow and the fast muscle fiber, which has less and more mitochondria, they adapt differently to different exercise training. And this is quite important because there are some evidences that shows that if you have more slow fiber, if you have more mitochondria in the muscle, you are less prone to get diabetes. So let me just see. I have some messages I was trying to read. Okay, then I'm gonna move to the different topic. So this is about mitochondrial supercomplexes. So those who study biochemistry, they must know what biochemistry. They must know what is mitochondria. So mitochondria, the basic function of mitochondria is to produce the energy in the form of a reaction called oxidative phosphorylation, and that is happening in the so-called complex 1 to 5 subunits in the mitochondria. So there is a theory that these mitochondrial subunits, they work individually or they work in the form of complexes. They form these so-called supercomplexes. And this is quite difficult to study in the lab because so far people have been isolating mitochondria from the tissues and separating them on the gel and doing some Western blot to check what complexes are changing. And this is what was done in one of these paper from Switzerland, where they recruit the people and like they isolated mitochondria from the muscle before and after exercise training, and they saw that there is a super complex formation. But this has some disadvantage because this technique is not sensitive. So we decided to do this by mass spec, because we saw that when you do exercise training, this not only one subunit but many different subunit, they respond to exercise. So these many different bar graphs here, they represent individual subunit of this OXPOS complex. And we recently developed technique that was published in the Cell report, where we isolated mitochondria, and we loaded those mitochondria on the gel and isolated individual complexes, and we present how the composition of this complex is changing in response to exercise training. So if you want more detail, you can study this paper. I'm just throwing this as an example because I want to show what proteomics can do, because without proteomics technology, you will not be able to study something like this. And right now we are doing another study where we are putting mice on the high fat diet, and we're also exercising them at the same time, meaning that we are making mice diabetic and at the same time we are making them exercise, and we want to see how exercise can use to treat the diabetes. And this graph here just shows that our protocol works very well. If you do the treadmill running, you can almost reverse majority of features of the diabetes. And this is also represented here with the tissue weight and stuff. And we are currently looking into mitochondrial supercomplex formation in this study. I'm going to switch gears now and talk about PTM proteomics. PTM means post-translation modification. So the proteins, we have many proteins in the body, right? So we have more like close to 20,000 proteins in the body. And the proteins are just not proteins. They get modified by addition of certain chemical moiety on it. And there are more than 200 known modifications on the protein. And one of the most widely studied modifications are this one that are listed here: phosphorylation, acetylation, methylation, ubiquitination, and the glycosylation. And you can study this protein modification by using proteomics. And I will give some of the examples of this modification. So we, together with one of the group here, recruited like 8 different individuals which underwent so-called high intensity interval exercise. And then after 5 weeks, we terminated the study and we took the muscle biopsies before and after. And these figures here just shows that if you do that kind of exercise, your mitochondrial performance is improved. Then we isolated proteins and we studied post-translation modification that is called as protein acetylation, because it's important for mitochondrial metabolism. So we did this workflow where we digested proteins and we measure proteome and acetylation using different computational pipelines. And we can see that if you look at the top 10 acetylated protein, they were mainly enzymes that are involved in the mitochondrial metabolism. But in addition to that, you also have a protein called histone. And this so-called volcano plot shows that there were huge changes in response to exercise, and it was mainly the mitochondrial protein. And this work was done in collaboration with a couple of professors at the University of Copenhagen. And just for you to know, you might have heard about this yo-yo test that we use in the cricket, right, in India. So you need to pass yo-yo test to get selected as in the Indian cricket team. And the yo-yo test was invented by this guy. His name is Jens Bangsbo. So I'm working with him here in Copenhagen. Okay, so then the next part, I will talk a little bit about the insulin-induced phosphoproteome. So you remember this slide earlier, when you have a diabetes, your insulin doesn't work. You have an impaired phosphorylation in this protein. That's why we are looking at the insulin-induced phosphoproteome in both in human and mice. So this is an example from the mice study where we injected insulin in the mice. We also have similar study in the diabetic in the human. So we isolated muscles and we look at the insulin-induced phosphoproteome. You see expected changes. Insulin changes phosphoproteome quite a lot. And then these are just numbers, how many phosphorylation sites we can detect. They were like 1,800, no, there are like 18,000 phosphosites that are changing in response to insulin. You don't need to think so much about these numbers. It just shows the distribution of different amino acid, how they are phosphorylated. And what we normally do with this kind of study is like we pick up certain candidates that are regulated with insulin, and we do the functional follow-up in the study. And in this study, we found that there is this protein called RAP10 that is involved in the trafficking of the GLUT4 here, and we are actually revealing some new biology. This was not known before, and we are currently following that up. It's unpublished data. And what we show is like this RAP10 protein is also involved in the Parkinson disease, and our functional follow-up studies shows that there could be a link between diabetes and the Parkinson's disease. Because if you see in the population study, people who have diabetes, they are more prone to get Parkinson's. And I think this mechanism that we have here can explain some of those aspects. And then on this slide, we just have a lot of signaling data that just explains the mechanism, how this protein is phosphorylated. But I won't go in detail because I don't think you guys are aware of Western blotting technique so much. So in the last part of my talk, I will talk about secretomics or proteomics of secreted proteins. So secreted proteins means the proteins that are circulating in our blood. We have different organs in the body, and depending on different environmental incidents, like like if you have a diabetes, cancer, or if you do exercise, different organs in the body, they secrete proteins and they go in the blood. And proteins can go from one organ to another organ, like protein released from the liver could act on muscle, protein released from muscle could act on the brain, and things like that. And we study that by proteomics. This is just a brief intro to what is secreted proteins. And the well-known example of secreted protein is insulin, FGF21, and GLP. And the way we study that is like we either analyze the blood or plasma or urine sample or the saliva sample, or sometimes we also take the microdialysis sample. But we also grow the cells in the plate, and then we treat them with certain drugs and collect the media, and we do the proteomic analysis on that. And this is how we study the secreted proteins. So one of the first study where we developed the method, we grow the muscle cells, and we made them insulin resistance by treating them with high lipid. And I collected the media, and by using bioinformatics and the proteomics pipeline, we identified more than 1,000 proteins, and of which 40% proteins were regulated by diabetic conditions. And that includes some well-known markers that are known in the field. We also did the proteome of human brown and the white fat, and I think everyone knows what fat is. What white fat is? It's around our belly, on our tummy. That's a bad fat, but we also have some fat in our neck area. That's called brown fat, and that's a good fat. So because this brown fat has a lot of mitochondria, it can burn a lot of energy. So we isolated cells from the human, from the white and the brown adipocytes, and grow them in the culture, collected the media, and by mass spec, we looked into what proteins are coming from the brown fat, because they are supposed to be the good protein. And this was in collaboration with the pharma company and the hospital here. And the goal here was to discover protein that comes from the brown fat, which could have some anti-diabetic effect. And indeed, we found many of them, and we published one of this protein where it's called EPDR1, which is specifically coming from the brown fat, and it has a potential role in the mitochondrial respiration. Yeah, so I just want to go back again to this previous slide where I mentioned that exercise has a beneficial effect on different organs. And there is a hypothesis in the field that when you do exercise, and exercise again could be walking, just walking, like fast walking, different organs in the body, they release secreted proteins in the plasma, and maybe they have a beneficial effect in the treatment of diabetes. And this is what we are studying. We do different model of exercise, and then we collect the sample. So it could be one leg exercise, and sample are collected from the arteries and the vein. It could be microdialysis sample, where the samples are collected in the extracellular space. We could contract the muscles in the plate using electricity, collect the samples. We can also exercise people and collect the plasma. And the goal here is actually to find out secreted proteins that are coming after exercise and whether they have any anti-diabetic effects. And we use the standard proteomic workflow for that. And once we discover those proteins, we do many different functional studies in different cell models to see whether there is an anti-diabetic effect for those proteins and peptides. And in one such example, we discover this protein that is released upon muscle contraction. And this graph shows the mitochondrial respiration. It increases the mitochondrial respiration. When we did the proteomic analysis, we could accurately sequence this protein. And then what we saw is, like, if you do exercise, this protein is increased in the mouse and in the diabetic patients. This NGT is normal patient and type 2 diabetic patients. Immediately after exercise, this protein goes up in our blood, and 3 hours exercise, it goes back to normal. So this tells us that, even if you have exercise, even if you have a diabetes, this protein goes up immediately after exercise. And the message here is like, exercise can work better than your medication, because you have a protein that is released and that has a little bit of like anti-diabetic effects. And here it just shows, like, doesn't matter what kind of exercise you do, this protein goes up immediately after exercise. I will skip this slide because it's very complex for you to understand. And what is this protein? So this protein is actually banned by doping agencies, like, in the sports science, athletes, they buy this protein, they because their exercise performance is increased, so it's used in the doping. And even though it's banned by doping, it's illegally used in the horse racing. So people are injecting horses with this protein so they can run faster. But for us, this protein was interesting because it has some anti-diabetic effect. And then we looked at what kind of effects it has in detail. So we show that, it increases the formation of bone. So it has a important role in bone metabolism. It forms new neurons. So it actually has a good effect on the neuron and your brain. And we also saw that it helps in developing muscle. So then we looked into the insulin secretion. It increases the insulin secretion in pancreas. It has a mild effect in treatment of obesity and diabetes. So we did this work in collaboration with the Novo Nordisk company. Again, I won't go in the experimental details here, but just want to show that this protein has an anti-diabetic effect. So last few slides. So we have ongoing studies because we believe that if you analyze the blood after exercise, there is a possibility of discovering proteins and peptides which have a anti-diabetic effect. So we did this study where we recruited 25 healthy individuals. We did their baseline screening. We put them on the exercise, and we have collected plasma, urine, and saliva at different time points. And we have analyzed like proteome and the metabolome in this case. I will not show any data on that because it's an ongoing study, but I will show some data. So in plasma or in the blood, it's very difficult to detect the protein. It just shows here the dynamic range, and it's because of the complexity of the samples. So what we are doing in this study is, like, we are taking alternate approach where we are getting rid of proteins and analyzing peptides. It's called peptidomics. But we are also isolating something called extracellular vesicle. These are small round vesicles which carries proteins from one organ to another organ. So it's a basically column-based approach. So we isolate those EVs or extracellular vesicle. We confirm them by cryo-EM that we have a pure preparation, or so-called this nanoparticle tracking. And then we look at their proteome, and this is covered by this grant, and also Lona and Diana from the group are working on this one. So this is a little bit more about the EV, what EVs are. So our extracellular vesicle, they are like kind of a cargo or packing. They mainly contain protein, but they also have DNA and RNAs. And one of our goal here is like to isolate the EVs from these different time points that we have in the study, and see what is the composition of this EV by using different omics technology, proteomics, transcriptomics, metabolomics, and lipidomics. And we also want to see what is the effect of those EVs on the mitochondrial respiration and, let's say, diabetic conditions. So this is our workflow again for EV. So we compared a couple of workflows that involve centrifugation and the isolation with the column. And in our hand, the column-based approach, which is at the bottom here, it works better. Centrifugation doesn't work better. And we did that because there is a controversy in the field which approach is better. And what we then we use this we use this mass spec, TIMS mass spec, and we identified more than 1,000 proteins. We did a downstream analysis, and we have expected markers that should be there. And what we saw, these are different time points of exercise. This is a PCA plot. We saw a lot of changes happening in the blood 3 hours after exercise. And we are looking into that. And I think most likely it's because body is recovering from the exercise bout, and you see a lot of changes 3-hour exercise after exercise. And we are looking into whether there are some anti-diabetic candidates here that we can synthesize. So this is my summary slide. So yeah, might be very complex for you, but if you know proteomics, you can apply it for all kinds of research question in the field of metabolism or any other research. Expression proteomics help identifying differentially regulated proteins. Mitochondrial supercomplex enables to study dynamics of supercomplex formation in the different condition. PTM proteomics identifies the novel regulator of cellular process, and secretomics allows you to identify novel secreted protein with the therapeutic potential. So with this, I would like to thank people that I work with and my group. So not everyone is in this picture, but yeah, so majority of them, they are there. So with that, I can take questions.
Hello.
Yeah.
Hello, all attendees. Please, Dr. Atul has completed his presentation. Now, if you have queries, you can ask him. Session is open for the discussion.
Hello, Atul.
Yeah.
If we exercise, then diabetes will be reduced.
Yes, I mean that's what the evidence says. Like, but it doesn't need to be. You need to exercise regularly. You cannot be only one exercise, right? So every day, 30 minutes, or in 1 week, it should be 150 minutes of physical activity.
So which one? Which one?
Anything, like you can walk, you can bike, cycling or walking, or early morning walking is also good.
As a researcher, what are you doing?
What?
What are you doing exercise?
Yeah, in my case, personally, I go to gym 3 times a week, 2 to 3 times a week, or I walk if I cannot go to gym.
Okay. Yes, anybody?
I would imagine that whatever research I'm doing, it's a very complex thing for audience here, because I know that very few institutes in India, they have access to mass spec, only top institute. And maybe the audience here, they might have very strong background in chemistry, but not in the molecular biology or proteomics. But nevertheless, I'm really happy to share what we do.
In India, nearly 50% people are suffering from diabetes.
Yeah, diabetes is a big problem in India, right? So I follow this very closely, and I think what I can mention here is like Indian diabetic phenotype is very different than the diabetes that we have in Europe. The thing is that in India, people can be lean, meaning that not very obese. Like you can be lean, you don't look like you're very big, but you have very severe diabetes. And people don't know what it is exactly, but I think for me probably it is related to our eating habit that are
मोरे
changing,
सर
right?
जरा 1 मिनिट इकडे मला फक्त कॉन्फरन्स आहे.
Hello, sir. Deshmukh, sir.
Bola.
I have one question. Sir,
Bola.
what is the relationship
Hello.
of aldose reductase enzyme with the diabetes?
I don't know that thing, but there are some studies that shows the mutation in that one has a high incidence of diabetes, but we don't see at the proteomic level, we don't see consistent regulation in aldolase enzyme. We do see aldose reductase. No, we don't see.
Hello.
Yeah.
I think there are no queries. There are no queries. Sir, I would like to ask one question. Why there is loss in weight in diabetic patient?
What do you mean loss in weight in diabetic patients? You always have, normally most of the time you have increased weight in the diabetic patients.
Because my wife was having weight about 60 or more than 60, but few months back sugar is detected and she has loss in weight. So what is
So
what could be the
that's
reason?
quite unusual because the weight gain and the obesity is strongly associated with the diabetes. In this particular case, there might be weight loss because there could be a different subtype of diabetes, because diabetes, even though is defined as one term, there are many subcategories, and those categories are very difficult to understand because there are certain protein signaling pathways that are affected in the one type of diabetes compared to another type of diabetes. That's why some people are resistant to certain drugs. They don't work because the mechanisms are not same. And this example that you are telling, weight reduction in diabetes, it's a rare thing. So I think it must be a different type of diabetes and it needs thorough investigation at the molecular level. Ideally, if you were in Europe, they will take the blood samples and they will do the DNA sequencing completely and see if there are mutation on any genes and if there is a specific type of diabetes. But the whole DNA sequencing is not common in India. They are
Thank you,
doing only
doctor.
for COVID in certain case.
Okay. Thank you, Dr. Atul. You have delivered very nice and informative lecture. Even though you are saying that the topic is complex, but you have made it very simple. As far as the diabetes and obesity are considered, you have given the introduction about these two types of diabetes. And in India, diabetes is spreading very fast, and told that out of 4 persons, 3 are suffering from the diabetes. So exercise is the best solution for reducing the diabetic conditions. Thank you, Dr. Atul, and you have given explanation on different types of proteomics, which is very helpful for understanding the concept. Even though for the general science students or researchers, the concept is complex, you have made it very simple and easier to understand. Really, we are very thankful to you on behalf of the organizers. Thank you very much, sir.
Thank you. I'm leaving the meeting now because I have another meeting. So thank you very much.
Okay.
Now I hand session to Dr. Kadam sir. So I requested to Dr. Kadam sir to continue next session.
Thank you, madam. Good afternoon to all the delegates and the participants of the International E-Conference on Proteomics Application to Biomedical Research 2022. Thank you for this overwhelming response to the conference. I am here as a session coordinator of validatory function. So now I would like to request Mr. R.S. Gaikwad, sir, head of the department, Vignan Mahadhyalaya, Sangola, to deliver the conference overview.
Good afternoon, everyone. I am Professor R.S. Gaikwad, convener of this conference on proteomic application to biomedical research 2022. We are here for the valedictory function of the conference. Now I would like to give conference overview. Day first includes inauguration of the conference. Firstly, Dr. S.S. Dasade introduced the chief guest of inaugural function, Professor G.B. Zore from SRT University School of Life Sciences, Nanded. He has delivered excellent keynote address, which includes all the essential aspects of the proteomics and its application to biomedical research. Also, he has nicely presented his research work in the concerned area. After this event, presidential address is given by Principal R.A. Phule. In his presidential address, he elucidated history of our Samstha, Sangola Taluka Shetkari Shikshan Prasarak Mandal and Vignan Mahavidyalaya Sangola. Also, Department of Chemistry striving for excellence in research and various educational activities. It is one of the leading departments in our college. The session was concluded by session coordinator J.V. Tombre sir. In session second, session coordinator Dr. S.N. Kadam introduced the chairman Professor S.B. Patwari, after which S.B. Patwari introduced the research person of invited talk, Professor K.D. Sonavne, very nicely. Professor K.D. Sonavne delivered excellent talk regarding his research related to proteomics. The session was concluded after discussion and vote of thanks. In session third, session coordinator introduced chairperson of the session, Professor S.S. Gaikwad, who introduced the research person of invited talk, Dr. Balaji Rokde from Ireland. In his talk, he delivered more beneficial talk regarding the research in the concerned area. In session 4, session coordinator A.R. Ingoli introduced chairperson, Dr. Lala Saheb Kashid, vice principal, Vidya Pratishthan, Baramati College. He inspected very preciously oral presentation of the participants, after which the session was conducted with vote of thanks. In session 5th, session coordinator Dr. J.V. Tombre introduced chairperson of the session, Dr. Devkar S.S., Head Department of Chemistry, SM College of Luz, who introduced today's chief guest, Dr. Dina Samedova from University of Copenhagen, Denmark, very nicely. In her talk, she delivered precious talk on mass spectrometry technology for proteomics and her research in concerned field. The session was concluded after discussion and vote of thanks by session coordinator. In session 5th, session coordinator Dr. Sheetal Shinde introduced to chairperson of the session, Dr. Madhusudan Bachute, principal, Sangola College, Sangola. He introduced chief guest, Dr. Atul Deshmukh, young scientist, University of Copenhagen, Denmark. In his precious, more valuable talks, he explains about functional proteomics in metabolism very nicely, which inspires researchers in this field. Then session was discussed and concluded after vote of thanks. Okay, thank you very much.
Now, participants of this conference in the portion there express the feedback about the conference. Thank you very much, sir. Now, may I request participant Professor Sanjay to express the feedback about the conference.
Good afternoon, everybody. I am very much thankful for giving me an opportunity to have interaction. Hello? Yes, I am audible?
Yes, yes.
Okay.
Yes, sir.
So I am very much thankful to you for giving me an opportunity to have an interaction as a participant in this international e-conference on proteomics applications to biomedical research organized by Vigyan Mahavidyalaya, Sangola. Actually, as a participant, I really impressed from total organization since yesterday and today's all the sessions for 2 days. Each resource person have delivered a very nice talk on their respective subjects. The chairperson of that every session have interacted and remarked well in their presidential address. In addition to that, total organization of this international conference we have enjoyed. There was no any online trouble, no any internet problem. And all the matter we have collected from this conference, including paper presentation, session, total organization. And I really appreciate the patience and efforts made by the convener, organizing secretary, and all their organizing member of this conference. So I request to management of this college to organize every year at least one like such a conference and motivate the students from research and give the opportunity for presentation of the paper. And I really appreciate the efforts made by you to publish the papers which will be published in UGC Research Journal. So again, as a participant, I am very much thankful to all the management of the college, organizing committee, all the resource person, convener, organizing secretary, member, and all of you. You again give such an opportunity every year for us. With this, I conclude. Thank you. Thank you very much.
Thank you, sir. Thank you very much for your precious feedback. Now, may I request another participants of the conference, Dr. Prakash Bansode, sir, to express the feedback about the conference. Dr. Bansode, sir. Dr. Prakash Bansode, sir, are you there? Okay, some technical problems may be there. Now, I would like to request Principal Dr. Bachuti, sir, to deliver the valedictory address to the conference.
Am I audible?
Over to Bachuti.
Kadam sir,
Yes.
am I audible?
Yes, sir.
Okay. Vigyan Mahavidyalaya Sangola has organized this 2 days e-conference on application of proteomics in biomedical research. On this occasion, the present members of the management, principal of the college, Dr. Unath Fude, convener of the conference, Dr. Professor Gaikwad, then his colleagues, then all the students, researchers, resource persons present. I again welcome you in valedictory function of this conference. At the outset, I congratulate the organizers for organizing this conference of 2 days, because organization of any event is a painful task, and online method has given us opportunity to organize these events easily, because the regular offline events require a lot of responsibilities. That is calling the resource persons, then their arrangement of lodging and boarding, then arrangement of participants of lodging and boarding. All these things have been eliminated in this online method, and therefore the task has been become somewhat easier. Though it has become easier, the task doesn't becomes more easy, because all the other formalities are to be done neatly and in time. And this organization of conference by Vidyan Mahavidyalaya Sangola is a good example of organization of the conference. Why such events are necessary to be organized? Because every year new students are coming in the colleges and in institutes. So they may be researchers, they may be students, or they may be the PhD holders or doing research for some other purpose. But what is happening in current date, or what is happening in every area, that student must know. And such platforms are very useful for them to get the knowledge about such events, whatever happening all over the world. Because we get information of new findings or new advancements in different fields. And therefore, organization of such events is very essential. Today and yesterday, Vidyan Mahavidyalaya Sangola has organized this conference to make available the platform for discussion on the very complex concept of proteomics. Because the general science students are not too much aware of these concepts, but even though these concepts are not too much knowing to general science students, but are very essential to know, because I have heard the 2 lectures today, one of Ms. Diana and another of Dr. Atul. And both these lectures have given us the importance of this proteomics, because Dr. Atul has stressed on importance of exercise in reducing the possibility of diabetes, or reducing the possibility of other disorders in our body. And therefore, such type of events are very useful. Not only that, the students and researchers get new ideas, what they can do further on this subject. These ideas get for the students. Nowadays, we have many problems which are in need of research. We require research on our water problem, that is its purification. We need the more work on the energy problem, fuel problem. All these problems need the attention, and all the researchers have to work on the local problems. That is whatever society needs, such problems must be taken into the consideration, and on such problems everyone must undertake the research. In the last week, there was a conference, national conference in Pune on the fuel. During the Ukraine and Russia war, there is shortage of fuel all over the world, and all world is looking for the alternative for the fuel, and everyone is researching on the fuels which are not the regular, that is petro fuels. There must be some alternative. Nowadays, the e-bikes are there, and these e-bikes require the lithium-based batteries. So, but these lithium-based batteries needs longer time for their charging, and after their life is over, there may be problem of their disposal. So on such problems, researchers must work and find the solutions, and therefore such type of events are very essential. So at the last moment, I congratulate again the organizers, Dr. Professor R.S. Gaikwad and his team for organizing such very nice event on the international level. And I am seeing that the response for this conference is also very nice. Papers have been presented, then the research persons have delivered the talks on very useful. Congratulate all the organizers, principal, then sir, over to you.
Thank you, sir. Thank you very much. Thank you very much, sir, for such marvelous valedictory address to the conference. Now, I will request Professor V.P. Ubali, sir, to convey the presidential remark. Professor Ubali, sir.
Sir, good evening. Hello, sir. Am I audible?
Yes, sir, you are audible.
Yes, sir.
Okay. Greetings from my side to all the participants and organizing committee members. I would like to say hello and good evening to one and all. First of all, I would like to take my opportunity to congratulate the Vijnan Mahavidyalaya Sangola for the nice, excellent organization of 2 days international e-conference on the topic Proteomics applications to biomedical research 2022. My friend, my colleague, and the person from the chemistry, Professor Madhusudan Bosete, has given the valedictory speech, which includes all things which are in connection with the conference and its outcome. After going through your deliberations and say brochure or a flyer, I noticed that the efforts taken by the management, as well as the convener, Mr. R.S. Gaikwad sir, then Ingole sir, then the acting principal Pule sir, the IQAC committee, Dasade sir, and all others has taken a long efforts for this successful organization. And the outcome of this conference is important with respect to the topic that there is a vast and emerging development in the biomedical sciences, and with respect to the chemistry also, maybe in connection with the sustained release or timely release of the medicines, then the use of the biomaterials which are made up of organic material, maybe the polymer, their adaptability by the body, and a n number of things are going there. And as a result of that, the college has selected the excellent topic, and for that purpose they invited the resource persons from the universities, one from Nanded and another from the go through also seen the one was from the Denmark also. The poster presentation and the deliberations are nice. The response given by the student is very good. In all, I can say the efforts taken by the college, and in such a period that is the particularly the corona has given the best opportunity to have such type of event in online mode also. And the college has used that technology, the Zoom applications, very nicely, and they tried at that level to have everything should go in smooth. Also, the convener and the principal has coordinated the eminent persons from our university itself. Also, the BOS chairman and the head of the departments of the different colleges were also invited to chair the session also. In all, the proper say management, organization, coordination is done very nice. And once again, I would like to say congratulations, and I appreciate the efforts taken by the college and the convener also. Congratulations to the convener R.S. Gaikwad sir, Ingole sir, principal Pule sir, and the resource persons also. And lastly, I would like to have the good words about the Professor Batsute. He is a excellent contribution in research and academics and in chemistry also. Thanks and congratulations to Madhusudan Batsute for his appreciating word also. Thanks to one and all. Thank you, sir.
Thank you very much, sir. Thank you for your precious presidential remark. Now, herein I request Dr. Shankar Dasade sir from Vigyan Mahavidyalaya Sangola to convey the vote of thanks.
Okay, sir, thank you. It is my great pleasure that I am here for vote of thanks. On behalf of my college and organizer of this international conference, I am very much grateful and thankful to President and Chief Guest of today's valedictory function. I am grateful and thankful to President, Secretary, and all directors of the institution. I am thankful to active principal of our college. I am thankful to all the resource persons within the country and outside the country for their excellent and beautiful presentation. Thankful to all the chairpersons who have chaired the 7 different sessions. Thankful to all foreign participants, delegates, all the research scholars from India and outside India. Special thanks to all my colleagues from my college and especially from Department of Chemistry and Department of Physics. We have taken more and more efforts in success of this international event. I am grateful to all students from our college, and it is our great honor that we are here with the eminent personality like esteemed Dr. Atul Deshmukh from Denmark. I am thankful to him for his excellent presentation. I am very much thankful and grateful to Dr. Daina for her very nice and excellent and valuable presentation. With the kind permission of President of today's valedictory function and with the consent of organizer of this international event, I declare that today's international event is over. Thank you one and all. Thank you so much. All the participants are requested to make on their videos.
Now, with the permission of the convener of the conference, I declare that the conference is over. The feedback link will be shared shortly to the participants.
Hello, Dasade sir.
Yes, sir.
Feedback link within short period पाठवताय ना?
Yeah.
Okay, thank you, sir.